Hello friends, now you are clear about the anatomy of external ear, middle ear and inner ear, isn't it? And also you know about the various receptors for hearing and equilibrium within the inner ear, right? So today we will be going into the very important topic of physiology of hearing, or how do you hear. Now what is the importance of hearing? It is one of the special senses, right? But it is getting less importance compared to our eyesight, isn't it? It is very often ignored by most people, and that is why Helen Keller rightly said, blindness cut you off from things and this deafness cut you off from people. Okay, so it is very important that we learn about hearing and we learn why the hearing loss occur and how this hearing loss can be treated. Only if we are able to know how the hearing occurs will we be able to understand why hearing loss is occurring in the diseased ear, what type of hearing loss is occurring, and how this hearing loss can be overcome. Okay, so we'll be going to the topic physiology of hearing, and this hearing can be broadly divided into three steps. Okay, first step is conduction of the sound waves, second step is transduction, and third step is the neural pathways. Now what is happening is conduction. In conduction, the sound waves from the external ear, okay, the sound waves are coming through the external ear, right? The pinna is acting like a funnel. It is connecting the sound waves and it is localizing it to the tympanic membrane, the small area on the tympanic membrane, and from there it is going to the middle ear and then to the inner ear. That is what is called conduction. From there, the sound waves are getting transformed into electrical energy, and that change in the form of energy is what is known as transduction. Okay, and this transducted sound waves, which have become electrical energy, are getting transmitted through the neural pathways. Okay, there are neural pathways going through the brain stem to finally reach the brain's auditory area. Okay, the superior temporal gyrus. Okay, to reach that you have to go through neural pathways. Okay, now what is the function of middle ear? This middle ear, is it just a conductive apparatus for sound wave to walk through like this? No, it is not a simple mechanism like this. Okay, the middle ear is not just providing a conductive apparatus. I will tell you in detail, but for now, what do you see here? You can see Shah Rukh Khan is taking a dip in the water, right? And when he is taking dip in the water, he is unable to hear what is being talked outside. Why? Because the sound waves speed is different in water and the air. Okay, and because of that difference in speed, you are unable to hear. So in our inner ear, you know that there is perilymph and endolymph, right? And so the sound waves are getting transmitted through the perilymph after reaching the inner ear. Okay, and so because of that, because of the change in impedance, the sound wave that is properly reaching the inner ear, the part before it gets transduced, it will be only 3%. Okay, only 3% of the sound wave is expected to reach, but the sound wave that is reaching is actually 50% of the sound waves are actually reaching. Okay, 50% of the energy is getting retained. Why is it so? Because of something known as impedance matching. Okay, impedance matching is done by the middle ear. Okay, it is the function of the middle ear, impedance matching. Now what is this impedance matching? What do you mean by impedance? Impedance is actually the opposite of conductance. Okay, and impedance, you can think it in terms of resistance. It is a resistance offered. Okay, so to overcome the resistance offered by the change in the medium, what is being done is the impedance is being matched, and that is done with the help of the middle ear ossicles. Okay, what are the ossicles within the middle ear? You know, we have got three ossicles, the malleus, incus and the stapes, and the malleus handle is actually connected to the pars tensa. It is between the middle fibrous and inner mucosal layer of the pars tensa, and when the sound waves reach the tympanic membrane, it is localized in the tympanic membrane by the funnel shaped action of the pinna, and this sound waves causes the vibration of the tympanic membrane, and that vibration results in vibration of the ear ossicles. Okay, when the ear ossicles vibrate, what happens? When the ear ossicle vibrates, the sound waves go into the oval window, and from there it goes through the scala vestibuli and comes out through the scala tympani through the round window. Okay, that is what is happening, and because of that there is a movement of the basilar membrane, and because of the movement of the basilar membrane there is shearing action of the organ of Cotte or the tectorial membrane, and because of that the sound is getting transmitted. I'll be going to the details soon. Okay, so how is this impedance matching occurring? There are three basic mechanisms for impedance matching. Okay, what are they? These midlayer ossicles, they are acting as transformers. Okay, transformers. Now what is this transformer? Is this a transformer of the Marvel? No. The transformers are structures which we use for increasing or decreasing the voltage, right? In our homes, for the transmission of the electrical energy, we have to increase or decrease the voltage. So this is being done by transformers. Similarly, to decrease the impedance cause, we use the midlayer ossicles. And how is this being done? Okay, there are three basic mechanisms within the midlayer. One is the catenary lever mechanism, 2 ossicular lever mechanism, and 3 the hydraulic lever mechanism. Okay, and for that our hammer, anvil, and the stirrup contributes. Okay, so first of all, what is this hydraulic lever action? Okay, you all know that the effective area of tympanic membrane is 55 millimeter square. I already told you the actual vibratory area is 90 millimeter square, but the effective vibratory area is only 55 millimeter square, and the foot plate, it is 3.2 millimeter square. So such a large difference is occurring in the vibratory area. Okay, so the partitions of the tympanic membrane, the effective vibratory area is 55 square meter, and onto it is attached the handle of malleus, and here you have the ingus attached, right? And on the ingus it is attached the head of stapes, and then you have the foot plate of stapes, it is attached to the oval window, right? So this is how it appears, and here you have the round window, and this round window forms a preferential pathway for the sound waves. Okay, so when the sound is going and hitting this area, that is the effective vibratory area of the tympanic membrane, the pressure remains the same, right? If the force is reaching a smaller area, will the force be magnified or getting reduced? What do you think? The pressure has to remain constant, right? Here the area is large and the force is small, and when it reaches here, what happens? The force increases magnificently. Okay, there is a profound increase in force. Okay, and this is ossicular lever mechanism. Okay, ossicular lever mechanism. Now what did Mr. Archimedes say? He said that if he is given a lever and a place to stand, he can even lift the earth with the lever. Okay, so that is the basic principle of a lever, and when applying the same principle to the case of our ossicles, okay, the ear ossicle handle of malleus is 1.3 times longer than the long process of ingus. Okay, long process of ingus. So this 1.3 times compared to the 17 times from the aerial ratio. Okay, what is the aerial ratio? 55 millimeter square divided by 3.2, that is equal to 17. Okay, 17 into 1.3. 1.3 is the lever ratio. Okay, that is the advantage obtained, and that is approximately 22 times. Okay, 22 times. Okay, So 22 times amplification is obtained because of that. Now there is one more effect. What is that? You have already told that there is something called the umbo, right? Because of the tending of the pars tensor like this. Okay, and here you know there is a curved membrane effect occurring. Okay, curved membrane effect. Why? Because TM when stretched exerts greater force upon points of attachment. As endless is immobile, sound energy is applied to TM is amplified at the area, at the central area. Okay, this is what is called the catenary lever mechanism. Okay, so here you have 2x amplification taking place. Okay, 2x amplification is taking place due to the sound lever mechanism. Okay, it is also known as curved membrane effect. Okay, curved membrane effect. Yes, so here the force will be maximum, that is 2x times. Okay, that is the basic principle of catenary lever or curved membrane effect. Okay, catenary lever or curved membrane effect. So these are the three mechanisms that contribute. The first is the hydraulic lever mechanism. Okay, what is the aerial ratio? It is 17 times. Now here you have 1.3 times and here 2 times. Okay, so all these mechanisms contribute to increasing the transmittance of sound. Okay, so it decreases the impedance and helps to match the impedance. Okay, so there is one more mechanism which helps in this, and what is that? It is the phase differential that is being maintained at the area of oval window and round window. There is a preferential pathway to the oval window because of the oscillator change. Okay, oscillator change, since sound travels maximum in solids, okay, compared to liquids and air, the sound waves which are going through the pars tensor will be transmitted through the ear ossicles rather than going through the air and going to the round window. Okay, so sound wave will be reaching the ear ossicles first. Okay, this is known as ossicular coupling, where the sound wave is going through the handle of malleus and reaching the oval window. Okay, so this is ossicular coupling happening here, and there is a preferential pathway through this. Okay, when sound wave is going and reaching the oval window, the sound wave, when it is in compression over here, it will be in rarefaction at the area of round window, or rather they both are always maintaining a phase differential. Okay, a phase differential is being maintained by the round window and the oval window. Okay, this difference in phase between the oval window and round window helps to establish the sound wave through the cochlea. Okay, and so these are the three main mechanisms. One is the catenary lever, second is ossicular lever, and then third is hydraulic lever, and apart from that you have the phase differential being maintained. Okay, all this contributes to the impedance matching. Acoustic reflex. What is this acoustic reflex? When the sound wave that is reaching the mid layer is too loud, you have to dampen it, right? Why? To protect the inner ear from noise induced hearing loss. It is similar to the transformers decreasing the load of voltage so as to prevent the problem due to the increased load. So both these are happening in the mid layer system, that is increasing the impedance to protect as well as decreasing the impedance to match. Okay, so this important function which helps to decrease the sound that is reaching the inner ear, that is what is known as the acoustic reflex. Okay, acoustic reflex, and it is mediated primarily by the stapedius muscle, and of course the tensor tympani muscle also contributes. Okay, tensor tympani muscle also contributes. Now what is happening here? The stapedius muscle pulls the stapes foot plate inward from the oval window. Okay, the stapes foot plate is attached to the oval window. It has to be pulled inward. okay, to decrease the sound waves transmitted through the inner ear, and that is being done by the stapedius muscle. Okay, now what is this tensor tympani muscle doing? Tensor tympani muscle is pulling the tympanic membrane inward, okay, and both these things increase the impedance of the ossicular system, and that helps in preventing a noise induced hearing loss. Okay, so what happens when the stapedius muscle is damaged, or when there is any damage to the facial nerve and the nerve to stapedius is no longer functional? What happens? There will be hyperacusis. This hyperacusis is occurring because of the damage to the nerve to stapedius. Okay, and that is why it occurs, because stapedius muscle contributes to decreasing or dampening the sound waves that are reaching the ear. Okay, so when a patient who is having a facial palsy, if the nerve to stapedius is also involved, then what happens? The patient, if he is exposed to loud sounds, he can get a noise induced hearing loss. Okay, because the important function of stapedius of dampening in the sound is lost. Okay, so now we know the two important functions of middle ear. One is the impedance matching, and second is the acoustic reflex. Okay, so second is the acoustic reflex, and the first is of course impedance matching. Okay, so these are the two major functions of middle ear, and hence middle ear does not provide a conduit alone, okay, for the transmittance of sound. It is not just providing a conduit for walking of the sound waves from the external ear to the inner ear. It is having the two major functions of impedance matching as well as acoustic reflex. Okay, now if there has been no middle ear, okay, if the sound is directly stimulating the inner ear, what is that known as? That is what is called the bone conduction. Okay, bone conduction. Now who is this guy? You might be familiar with this music, right? Yes, very soothing to our ears, right? This music. This is the famous music by the famous musician. Who is this? He is someone who became deaf in his later life. It is said that some of his greatest melodies have been created after he became deaf. Okay, so he is a real legend, right? And who is that? Is none other than Beethoven. Okay, Beethoven. And what did this Beethoven do? He used to put some stick to his teeth, and with that he used to attach it to the piano so as to hear the piano better. Okay, now what is he utilizing here? He is utilizing the bone conduction. Okay, bone conduction. If you have read the book Origin by Dan Brown, you might be familiar with this. Okay, this is a sentence from the book. Here he says the bone conduction technology was in fact not invented by Mr. Kirsch. It was actually invented by Ludwig von Beethoven in the 18th century. Okay, and with the metal rod attached to the piano, through the vibrations of his jawbone he is able to hear better. Okay, because he was having an otosclerosis which was causing a conductive hearing loss. Okay, so that is the importance of knowing bone conduction. Now what does this bone conduction indicate? Bone conduction has got three pathways. Okay, what are these three pathways? It can directly go and stimulate the cochlea like this. Okay, when you are giving a sound over the temporal bone, it can directly stimulate the cochlea like this, or it can go through the ear ossicles and reach the cochlea, or it can go through the external auditory canal and reach. Now what is the significance of this? You might have heard that in otosclerosis there is something known as Carhart's notch, right? Carhart's notch is seen at the area of 2 kHz in the bone conduction curve. Why is it occurring? It is precisely because of this reason. Why? Because when you are having various frequencies going through the sound wave, the natural resonance frequencies are seen only in certain areas. Okay, the area of axonotary canal has got the natural resonant frequency at 3000 Hz. Tympanic membrane has it at 800 to 1600 Hz. Midlear at 800 Hz and ear ossicles at 500 to 2000 Hz. Okay, ear ossicle at 500 to 2000 Hz. And because of this, what happens is that at the area of 2000 Hz, there will be a dip when you are having a fixation of stapes footplate, as in autosclerosis. Okay, in autosclerosis, when the stapes footplate get fixed, there is a dip in the bone conduction curve, and that is what is called the Carhart's notch. Carhart's notch. The reason is because there are three pathways of bone conduction, and the second pathway, that is through the ear ossicle, is getting affected here. Okay, whereas the pathway through the axonotary canal is not at all affected here. Okay, so that is the reason why it occurs like this. But usually the bone conduction is always considered as a measurement of the cochlear reserve. Okay, cochlear reserve. And when you have a bone conduction problem, it means it is a problem with the sensory neural pathway. Okay, it means it is a sensory neural hearing loss. Okay, only when the air conduction pathway is affected, then it means it is a conductive deafness. Okay, conductive deafness. Okay, for now just understand that bone conduction is an estimate of the cochlear reserve, and if there is a problem with bone conduction, it means the sensory neural hearing loss is seen in this patient. Okay, now what happens when the sound waves reaches the inner ear? After reaching the inner ear, what is happening to the sound waves? Now we know the cross section of the cochlea, it is like this, right? It appears like a snail, and when you take the cross section, you have a scala vestibuli above and a scala tympani below, and in between you have a scala media, and on the scala media you have the organ of Corti. Okay, organ of Corti is named after the famous Alfonso Corti. Okay, Alfonso Corti is an Italian scientist who discovered the sensory organ for hearing. There is the organ of Corti. Okay, now this organ of Corti consists of the basilar membrane, on which you have the tectorial membrane, and within that you have the inner ear cell in one row and outer ear cells in 3 rows, and there are various supporting cells such as the Clodius cells, Henson cells. Okay, all these are supporting cells. Now what is happening when the sound waves reaches the inner ear? As you can see here, the cochlea and the labyrinth is seen within the inner strand of the petrous part of the temporal bone, and if you dissect it out, you will be able to see that the cochlea, which is seen on the anterior part of the bony labyrinth, it is seen anterior to the vestibule, right? And the vestibule has got the oval window. When the footplate of stapes is getting vibrated, the oval window, through the oval window, the sound gets transmitted to the perilymph, and the perilymph vibrates, the scala vestibuli vibrates, and then the scala tympani vibrates, and because of that there is a shearing action taking place in the basilar membrane, and because of that shearing action, the hair cells move over the tectorial membrane, which is forming a gelatinous cover over that area. Okay, as you can see in this picture. Now how does transduction happen? There is a shearing force between the tectorial membrane and the inner hair cells, and these inner hair cells are the one which contribute major to the sensory neural signals. Okay, the outer hair cells are only having a nephron modulatory action, and because of that cochlear microphonics occur. Okay, so what is happening here? There is a bending of stereocilia that happens here, and because of that there is depolarization and opening of the calcium channels. Why this occurs? Because between the two stereocilia, which are of different heights, there are some tip links. Okay, there are tip links which are connecting the two stereocilia, and when they move apart, what happens? The stretch sensitive mechanical gated transduction channels open, and because of that potassium enters within, and because of the opening of the potassium, there is entering of the calcium channels. Okay, because of the opening of the stretch sensitive potassium channels, potassium enters, and because of the entry of the potassium, potassium sensitive calcium channels open, and because of that the neurotransmitters are released. What are these neurotransmitters? Glutamate is the neurotransmitter that is released. Okay, glutamate is the neurotransmitter that is released, and it initiates action potential in the auditory nerve. Okay, so this is how transduction occurs. Okay, so you can see how the hair cells have stereocilia on it. Okay, and when the hair cells stretch, what is happening? There is influx of sodium, and because of the influx of potassium, and because of the influx of potassium, what is happening? The calcium channels open, and because of the opening of the calcium channels, the calcium channels causes the neurotransmitter vesicles go and get released. Okay, so this is the tip links that you see here. Okay, tip links, and they get stretched, and because of the stretching, the stretch sensitive potassium channels open. And because of that, what happens? The voltage gated calcium channels open. Okay, because of that, calcium channels open, and because of the opening of the calcium channels, what happens? The neurotransmitters get released. The neurotransmitters are released. Okay, so that's about the transduction mechanism. Okay, now what are the various theories of hearing? The theories of hearing can be asked in your MCQ less often, but it is more of asked in your theory exams. Okay, theory exams. There are 4 basic theories: place theory by Helmholtz, telephone theory by Rutherford, volley theory by Weber, and traveling wave theory by the Becchesi. Okay, Becchesi. Now, the place theory and the telephone theory and volley theory, all these 3 are outdated. It is the traveling wave theory of Becchesi which is now in place. Okay, all other theory are only of historical importance. Okay, and von Becchesi got Nobel Prize for the discovery of this theory, the traveling wave theory. What does he say? When foot plate move inwards, it causes an elastic tension that build up in the basement membrane, which initiates a wave that travels along the basement membrane towards the helicotrema, and this wave has got a specialty. Okay, when the wavelength is less, that is the frequency is high, only the basal area gets stimulated. As you can see here, the wave is traveling only till the basal area. Okay, you can see how the cochlea, which is actually forming 2 and 3/4 turns around the modulus. It has been stretched for understanding purpose. Okay, and in this figure you can see that when a sound wave which is of high frequency is going, it is going and stimulating only the basal area. Okay, the apical area is not getting stimulated, only the basal area is stimulated. When a low frequency is given, it is going and stimulating the apical area. Okay, apical area. So this is what a traveling wave does. A traveling wave that is released because of the movement of the basement membrane, it goes and stimulates the basal area when it is of high frequency, and the apical area when it is of low frequency. Okay, now the other outdated theories were like the place theory said that there are certain specific areas of stimulation of the frequencies of sound waves, and the other theory, volley theory, said the sound waves are getting transmitted in volleys, and there are the sound is increasing according to the number of nerve fibers getting stimulated. All those theories are now outdated. Now just focus on that traveling wave theory, but you have to know the names of the other theories. Okay, because it may be asked for your theory exam as a 1 mark question. Okay, now what is this endocochlear potential? Now in the scala media you have something known as stria vascularis, right? If this is the basilar membrane, this is the tectorial membrane, and you have the stria vascularis here, right? Stria vascularis, and these stria vascularis, they have sodium potassium ATPase pump on this stria vascularis, and it is this triovascularis with the help of sodium potassium ATPase pump, which forms a source of energy for cochlear transduction, it causes endocochlear potential to occur because of the release of potassium into the endolymph. Okay, endolymph. So the endolymph has got a potential of plus 80 millivolt because of this endocochlear potential. Okay, and because of this channel here, the sodium potassium ATPase and the sodium potassium 2 chloride channel. Okay, you are also having a sodium potassium 2 chloride channel here, and because of that, when you are giving a loop diuretic, okay, loop diuretic which acts at the sodium potassium 2 chloride channel, okay, what happens when a loop diuretic such as furosemide is given? It goes and affects this sodium potassium chloride channel in addition to the sodium potassium chloride channel of the kidney, and because of that, what happens? Because of that, there will be a sensory neural hearing loss. Okay, a sensory neural hearing loss can affect by giving a furosemide because of this reason. Okay, that's about endocochlear potential. Now coming to the auditory neural pathway. 95% of the auditory fibers of spiral ganglion innervate the inner ear cell. Only the 5% innervate the outer ear cell, and these outer ear cells are having most of them as efferent fibers. Efferent fibers, okay, and this efferent fibers are to modulate the inner ear cell, and for modulating it, signals are coming from the auditory cortex. Okay, efferent pathway is occurring to modulate the inner ear cell. Okay, now east cochlear innervate both sides of the brain. How does this happen? From the 8th cranial nerve, where does the sound go to? It goes to the cochlear nucleus, right? After going to the dorsal and ventral cochlear nucleus, then it goes to the superior olivaric complex. Okay, so superior olivaric complex where it gets decussated, and the superior olivaric complex has got two important functions. What are they? One is it acts as a relay channel of auditory reflex. Second thing is it helps in sound localization. Okay, sound localization. From the superior olivaric complex, it goes into the lateral meniscus, and then it goes into the inferior colliculus, and then to the thalamus. Thalamus, medial geniculate body. Okay, medial geniculate body of thalamus. The lateral geniculate body is associated with the visual pathway, right? And from this medial geniculate body, it goes into the superior temporal gyrus or the Hirschel's gyrus. Okay, superior temporal gyrus or the Hirschel gyrus. So this is how you can memorize it. Okay, first you have the 8th nerve, and then you have the cochlear nucleus, then you have the superior olivaric complex. Superior olivaric complex is O. Okay, O. E. coli. And then you have the nucleus of the lateral meniscus. Nucleus of lateral meniscus is L. Then you have the inferior colliculus, medial geniculate body, and finally auditory cortex. Okay, auditory cortex, superior temporal gyrus or Hirschel's gyrus. Okay, this is the area 41 Brodmann area. Okay, so this is how sound wave travels. That is, initially it goes through the 8th nerve, then it reaches the dorsal and ventral cochlear nucleus. Okay, this is E, this is C. Then it goes to the olive or superior olivaric complex. Then you have the lateral meniscus. Then you have the inferior colliculus. Then you have the medial geniculate body. And then finally, after reaching the superior temporal gyrus or the Hirschel's gyrus, the auditory pathway is completed. Okay, auditory pathway is completed. So this is what is called equally ma for you to easily memorize. Now in the equally ma, the important thing that you remember is the superior aerobic complex has got two functions. What are they? One is it is the relay station for acoustic reflex, and the second thing is it helps for sound localization. Sound localization. How does it help in sound localization? By using the intraoral time clues. Okay, intraoral time clues between the two ears are taken, and here there is integration taking place. Okay, decussation is also taking place here. Because of that, superior auditory complex will be able to tell exactly where the sound waves are, where the sound waves are coming from, and it will know the location from which sound is heard. Okay, so broadband area 41 is the auditory cortex, and auditory association area is broadband area 42 and 22. Okay, this is a part of the Wernicke's area. Part of Wernicke's area. Okay, so this is how it appears when you take a functional imaging of the brain, that is fMRI. If you take, it will appear kind of like this. Okay, functional MRI, that is when a patient is hearing a sound or a music, and if you are taking a functional MRI like this, you can see these areas getting stimulated, that is the areas of the auditory pathway, that is E. coli ma. Okay, so that's about the physiology of hearing. So what are the things that we learned today? First of all, we learned that for hearing you need the middle ear impedance matching. Middle ear has got a very important role, that is in impedance matching, as well as acoustic reflex. That is the first thing we learned today. And then we learned that apart from conduction through the middle ear, you need to have transduction, which occurs at the inner ear. Okay, and for impedance matching we learned the three mechanisms, that is the hydraulic lever mechanism, the catenary lever, and the ossicular lever mechanism. And also the phase difference being provided helps with preferential pathway through the ossicular chain. Okay, and how does this transduction occur? Transduction occur because of the movement of the hair cells, movement of the hair cells over the tectorial membrane. Okay, hair cells over tectorial membrane can result in transduction, and after transduction what happens? It goes into the auditory pathway. Auditory pathway. And what is this auditory pathway? It is E. coli ma. Okay, E. coli ma. So this is the physiology of hearing in a nutshell. Please go and read this today itself. It is very important that you understand the mechanism of hearing, because when you are going into the diseases of the middle ear, you will realize how the conductive pathway is getting affected, and because of that there is problem with conduction of hearing and resulting in conductive hearing loss. Okay, and for understanding why conductive hearing loss occurs, why some type of conductive hearing loss is more when there is a ossicular discontinuity without a perforation is more than a ossicular discontinuity with perforation. All these things, if you are you will be learning in later classes, but for you to understand all that, you will have to have a thorough understanding of the physiology of hearing. Okay, so please go and learn today itself. Okay, thank you.